Hello, can anybody hear me?
We can hear you loud and clear. Just relax and wait.
Okay, thank you very much, bro.
Hello, sir. Hello, hello, hello. Hello, hello. Hello. Hello, hello, hello. No. Hello, hello, hello. 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 Hello.
Eh? Investigation. Oh, we are live streaming. What am I going to project for you? I'm going to go. So, I'm supposed to talk. Mr. Ebami, you want to come and sit down here? Uh, okay. Elemo, I'm right. Hold on now. Come and sit down here. Manage Zoom. Oh, there's something we do now. Once his camera is on, you something is not right.
Yeah. Can we next game?
One. Out of 10, we have out of 10. Don't have to count. Yes, yes. So the class is completed. But not in this case. But we are filling in.
I have joined the class, so that's that.
Okay, that is here. All right. Right here, we will online and they are teaching.
Yeah, I just did.
Come back to the phone. No, he has no phone. Forget to use for police. That was there. Wow. Now, another answer. Are you sharing this one? I mean,
Shouldn't pay.
You don't want to get to that.
Oh, yeah.
I'm ready. found. And with time, that because they did more than just looking for it. Triangulation, it tells you the might also become a serology. I said see liquid from now this is most useful
knife. People out of
think I'll just stand here to make it. Listen, listen, listen. Yeah. All right. Good morning, everyone. Um, Standing on all existing protocols. My name is Adeyemi Daniel Adetime, as Professor Adedeji rightly introduced me to you all. And we'll be talking about forensic entomology. And this talk is in collaboration with the resident forensic entomologists and experts in the Lagos State University, Dr. Alafia, that's seated here. So um, I am a visiting scientist in a laboratory, and I had my BSc from the University of Benin. I did zoology, but they call it animal and environmental biology there. And then I proceeded to the University of Cape Town in South Africa, where I obtained my PhD in forensic entomology. So this talk is largely an overview around what forensic entomology entails. And of course, this is a full-blown um, area in forensic science. And a day's talk alone is not sufficient to, of course, cover the broad area of forensic entomology. So this is just to give you all an overview of what forensic entomology is. And of course, we'll see some interesting things on the slides. And I'm more than happy to hear from you all, questions, comments. And I'm sure most of you all here may be used to seeing some gory things or weird things. So please bear with me. Just a word of caution if you're not used to seeing some weird or scary things. But I think we try to make the slide user friendly so that you can understand. So I'll be jumping in now. So sorry for the break in transmission. So I think I'm not sure with the technical setup, but we'll try our best to make this as friendly, user friendly as possible. So um, all right. So like I said earlier, this is more or less like an introductory to forensic entomology, and we'll be looking at various aspects. So for this talk or for this presentation, we sort of decided to put it into 4 key objectives that we hope to achieve at the end of this talk. So the first objective is I want you all to be able to appreciate the meaning and history of forensic entomology, and I hope you all will understand basic concepts with respect to time since death estimations. And thirdly, I want you all to familiarize yourself with the ecological categories of carrion, associated insects, and basic insect taxonomy. And lastly, estimations. All right. So generally, forensic entomology is the branch of forensic science that capitalizes on the biology and ecology of insects in clarifying specific questions in the civil, medical, and legal cases in the court of law. So it is a branch of forensic science. It is highly specialized, meaning that you require specialized training to be able to use insects in trying to sort of understand or apply insect life cycle in disentangling or trying to unravel cases that are perhaps civil in nature, medical in nature, or legal in nature. So generally, forensic entomology is divided into three broad categories. So we've got what they call the stored product pest forensic entomology. We've got the urban forensic entomology, and we've got the medico-legal forensic entomology, which is also known as the medico-criminal forensic entomology. So for the stored product pest forensic entomology, this is generally the category or branch of forensic entomology that has to do with the use of insects in trying to resolve disputes related to the presence of insects in food products, and in food production sites. For example, I went to one of the popular stores in South Africa to buy a chicken, this grilled chicken, because of course I was too lazy to cook. So I went to buy this grilled chicken. I won't mention the name of the store for safety reasons. And when I got the chicken, prior to me leaving the store, I was prompted to just check the box. And then when I looked through the transparent opening, window on the box, I realized that I saw several flies. Several flies were already in the box. So this aspect of forensic entomology could also be applied to situations whereby you go to the market, you buy a bag of beans. Then you get home and you realize that most of the beans are already infested with beans beetle or weevil, Callosobruchus maculatus. So the question, it begs the question, are you the one that sort of, or was it in your environment that sort of led to the infestation of the beans, or that was how you bought it from the store or from the market? So this aspect of forensic entomology focuses on the use of insects in trying to solve issues that has to do with the presence of insects in stored product or stored foods. And also you could see sometimes we've got our rice, bag of rice at home, we sometimes see weevils. So a forensic entomologist will come here to try to understand by looking at the life stages. Sometimes you see the adult stages, they're already flying about once you open the bag or once you open the container in which the beans or the rice cereals are being stored. So the forensic entomologist would try to say who is responsible. Is it that the infestation came in when you were sort of storing the food in your house, or is it that that was how you bought it from the store? So I could get home and say, no, I didn't buy these beans like this. It came with the insect from the store. So you can take it back to the store. And then the store owner will tell you, Oga, no be like this, you buy this thing, or these insects did not originate from the store. So a forensic entomologist in that case, by looking at the life cycle of the insects that are present in the stored products, food or whatever, they can try to deduce who is responsible. Is it you or the store owner or the store itself? And who is responsible. And then urban forensic entomology. This is the branch of forensic entomology that entails the use of insects in trying to find solutions to civil actions concerning insect infestations in human associated dwellings, buildings, and structures. So this is one of the most interesting parts. And a typical example of where a forensic entomologist will come in this category of forensic entomology is if you get into an apartment. Let's say you rent an apartment and you realize that there are bed bug infestation. What do? Obviously, you spend tons of money to exterminate the bed bugs, but then you expect that before you get into that apartment, the what is it called? The caretaker and everybody should have solved the bed bug infestation problem. But in this case, you go in and you realize that I'm sleeping, something is biting me, my body is itching. And then you look around the house and then you see that, oh my God, there are loads of bed bugs in this house. So the question that is of forensic importance here is who brought the bed bugs. Did you bring the bed bugs with your chairs and your beddings and your clothing, or the bed bugs were already in the apartment? Because bed bugs are insects that are mobile, they cling onto your clothes and they breed, and obviously they move around. So it begs the question, who is responsible? So in this case, a forensic entomologist who is also trained or skilled in other aspects of entomology can look at the developmental stages or the ages of the bed bugs in the house or in the apartment and try to disentangle. Oh, it seems like these bed bugs have been here for a long period of time. So there's no way these new occupants that came here 2 months ago or a month ago could have brought about this massive amount of bed bugs that could have led to this massive level of infestation. So this aspect of forensic entomology deals with the use of insects in finding solutions to civil cases such as bedbug infestation. I've heard of cases also where people buy a full building, a full house, and then they realize that all the wooden cupboards and everything, they've got massive termite infestation. So who is going to be responsible? I've paid you for the house. I expect that everything in the house should be proper, should be top notch. And if there's no formal disclosure prior to the purchase of the house or the property that there is bed bug, there is termite infestation, then I can take you to court and say, you will be responsible for exterminating this insect, or you give me back my money, because I do not want to deal with any bed bug issue or any termite issue. So this aspect of forensic entomology is what looks at those sort of situations. And no, just take it back to the previous slide. And lastly, what I just want to talk about this, because I'm not sure there are any cases in Nigeria, but there are cases elsewhere globally where you take your old one, let's say your babies or your old grandma or your old grandma to the hospital or to care homes. And maybe due to some neglect, maybe they pooed on their body or they peed on their body, and of course they couldn't wash up because the person that is taking care of them or the nurses or the doctors, they were not sensitive enough to be aware that there's a poo or there's a pee. So insects generally are attracted to the smell of the pee, the smell of the poo. So they come there, they lay their eggs. In fact, there are some people that cannot move. They are that old. And sometimes they sleep and they open their mouth. So insects generally, they are attracted to body openings, orifices, such as the eyes, the nose, and the mouth. So somebody that is not able to move due to age-related constraints, and the insect lay their eggs, and then, of course, you, their loved one, you get into the care home or the hospital, and then you see maggots coming out from the mouth of your loved one. Please tell me how you feel. Will you be happy? Of course not. So you can then ask or sue the hospital management or the care home or the caregiver or the nurse to say, hey, what are you doing? You're not doing your job properly. And then that could lead to maybe compensation, because also it could lead to infection and your loved one could pass away. So these are situations where you need the forensic entomologist. So the forensic can look at the insect and say, oh, it seems like this insect originated within the hospital or care home or such environment, and they can help you to establish a formal case that is reasonable in front of a judge or in the court of law. And then this last category of forensic entomology is more or less the most popular or the most widely researched area of forensic entomology. And this aspect of forensic entomology focuses on the use of insects in trying to deduce the circumstances surrounding the demise of an individual, especially in cases suspected to be homicides, suicides, or unnatural death. And like I said, it is the central focus of most forensic entomology. And generally, what we do here is to try to estimate the minimum time a person has passed on, the minimum time a person has died. So this branch of forensic entomology is also commonly known as medical criminal forensic entomology. So sometimes you see it in published papers, in textbooks, you see medical legal forensic entomology, or you see medical criminal forensic entomology. Forensic entomology does not only cover the three aspects. There are other interesting aspects, and I think Prof also highlighted some of them. So we do use insects to determine the duration of neglect of humans or animals. There are cases of cruelty where somebody is maltreating an animal. You're not giving them food, they're sick, they've got injuries, and then there are insects infesting their wound sites, or you're not taking care of humans. So by looking at the life cycle of the insect on the human body or the animal's body, you could try to deduce if the person has been neglected. One of the common cases here are babies. There are some people that will leave their babies on unwatched or unsupervised. And of course, there's accumulation of pee, there's accumulation of poo, and there's insect infestations, and it's just making the baby uncomfortable. There are cases where the baby could also have passed away due to the insect feeding on the body. And of course, babies cannot undo their pants and obviously wash themselves. So we sometimes use insects to deduce how long a person has been neglected or an animal has been neglected. Other examples include verification of cadaver relocation. So that means that, let's assume in LASU here, hopefully not, in LASU here, somebody is killed, and then the body is being relocated from LASU to somewhere like Ibadan. So generally, some studies have shown that within a given location or region, the insect community varies. So just like we've got in the Southwest, majority of us are Yorubas, etc. And then in the Southeast, we've got Igbos and all of that. So to a very large extent, some studies have shown that the community, the diversity of insects in a given environment varies from what is obtainable in another environment. So if you look at the deceased body and you realize that the insect community on that deceased body does not align with what has been documented in that environment, one could suspect that the body may have been relocated from one given environment to another. And I think I'll touch a bit on that later. And then thirdly, we've got prefers buried carcasses called the coffin flat. That's not, they are found on buried bodies, bodies buried in shallow graves, like maybe less than 1 meter, 20 centimeter, all of that. So insects could be used to determine if a body was exposed previously and was now buried, or was buried previously and was now exposed. Because some people, they may kill someone and, of course, bury the body in an environment. And when they realize that there are lots of leads and people are closing in and exhume the body and take the body to a different location. So insects that are sort of used to those kind of environments can be used to lead us as traces to unraveling the situation. And we've got post-submergence interval. So when somebody is being drowned or when person dies by drowning, there are some insects that are in the water that are attracted to the bodies. Some of them include Chironomidae and some other insect groups. So these insects are often in the water body, and by virtue of their association with these bodies, we can deduce the person was on land. And there are some insects that are amphibious. They can colonize the body, be it on land and of course in the water or at the surface of the water. So we use insect or forensic entomology to try to distinguish that kind of cases. And like Prof said, in the cases of poisoning or intoxication, so generally when an insect arrives on your body, they deposit their eggs or their maggots, those whitish things you see crawling around your rotten foods and all of that. So as they feed on your body, depending on the site of the body where they feed on, they could ingest some of these toxins or chemicals. And by the analysis of the maggots that you collect, bioactive compound. So if it is case of someone has taken sniper, for example, which is fairly common in Nigeria, or there is drug overdose, or someone has injected an unreasonable, then you could help to deduce, oh, it seems like this person was intoxicated, or the criminal investigator can then get some reasonable leads that can help to unravel what has brought about the demise of that person. And then there's wildlife poaching. This is very common in South Africa. I think in Nigeria, it's where people, of course, kill some animals like tusk, pangolin, their scales or their shells, and these are very expensive items. And insect can be used to help track this or estimate the exact time. But as forensic entomologists, we are recommended or advised to say minimum postmortem interval. So we are trying to give you a minimum time because it could be that there are some factors that may prevent insects from accessing the body. So instead of us saying postmortem interval, we say minimum postmortem interval. So I would just like to use that distinction. And then there's another terminology called the pre-appearance interval. So this is defined as the interval or the time between the death of an individual or animal until the first appearance of an insect taxon or life stage. So this could be an adult fly moving around. This could be the maggot or the larvae. So why this is important is because in some seasons, for example, whenever it's raining, most times insects are not active. So if a body is deposited out there whenever it's raining, there might not be active insects flying around. So how do we know when? So what we do most times is we do research, we do studies to at least get a sense of what happens in an environment. So if we know how long it takes for a particular insect group to arrive on a body, we can then use that in a situation whereby we see that same insect group on a deceased body in that specific environment. So what that means is that we cannot use data from Kaduna or from Kano to try to disentangle murder cases or deceased bodies in Lagos because there are different weather conditions, they've got different rainfall patterns, probably they've got different insect communities. So using data from a different region in this location would give you data information that is useless. And if you present that if you against a skilled lawyer, then they will tear you apart in the court of law. So it's important we know that. So moving on to the sort of ecological categories or groups of insects we often find on deceased bodies. So the first group of insects we often find, they are called necrophages. So these are insect groups that feed on the flesh of the body. An example includes your blowflies, like you can see there, your flesh flies, the one with the 3 stripes, and your beetle. So these are insects that feed on the flesh. They consume the flesh of the body. So they are called necrophages. So they are called necrophages. So these are insects that consume the flesh of the body. So the second group, they are called predators or parasites. So these are insect groups that feed on the adult and immature stages of other insects in and around the body. So examples include beetles, praying mantis, wasps, and ants. So these insect groups, they often do not feed on other insect groups that are in association with the body. And then we've got the omnivores. As we all know, man, we are omnivorous animals. We eat both flesh and plants. So there are some insect groups that feed on the flesh of the vertebrates or of the body, and of course they feed on other insects. So typical examples, we've got ants, we've got beetles, and we've got some specific wasps. And lastly, there are some insect groups that are called adventitives or they are incidentals. So these insect groups, they utilize the body as an extension of their environment. So sometimes we find cockroaches. We find cockroaches around the body, and we find butterflies, we find moths. So it's just, they use the body as an extension of their environment and they are just moving around. And sometimes I've read where cockroaches sometimes feed on the flesh of the body. So how do we use insects in PMI minimum estimation? So how do we use insects to estimate the minimum time a person has died? So, like I said earlier, the accurate estimation of the time since death is crucial in any sort of medical legal investigation. And there's no single scientifically proven method exists that can accurately and precisely estimate the time since death of a deceased person. So insects are often the most accurate and reliable method in PMI minimum estimation, especially after 48 to 72 hours of death. So when a person dies, most times on the first day or within hours, we start to see some degree of stiffening. So we see rigor mortis, algor mortis, which is the drop in body temperature, or livor mortis, where we see the cooling or the accumulation of body along certain parts of the body. But after 2 days, 48 hours, 72 hours, and so on. Those mortices, the algomortis, the stiffening of the hands or the limbs would not be used or cannot be used by forensic pathologists. So there is a consensus to a very large extent that insects are the most reliable sort of cues or parameters to estimate the death or times its death of a body, especially after 48 to 72 hours. So there are generally two complementary methods wherein insects can be used in estimating the minimum time a person has died. So firstly, we look at the developmental stage of the insect that is found on the body or around the body. And secondly, we look at the pattern of succession. So I would explain this in subsequent slides. So this is the picture of a decomposing pig. This picture was from my PhD research. So generally in the forensic scientific literature or in the forensic entomology or taphonomy literature, in Nigeria, and I think in most African countries, it is illegal to use human bodies for our kind of research. Although in the medical school, they use human bodies to teach anatomy, physiology, and all of that. But for our kind of research, at least in South Africa, where I did my research, and I think in Nigeria also, we cannot just take out a human corpse from maybe the morgue and put it out there in LASU somewhere and start. Obviously, if somebody sees that, they will probably run away or they will no longer come to LASU again. I don't know. But pigs are regarded as the most suitable alternative or surrogate to human bodies. And the reason is that we've got more or less similar integumentary system, we've got similar circulatory system, and we've got similar digestive system. So this makes them appropriate surrogates or proxies to human bodies. That's why we often use them. So this is a 60 kilogram, I think, female pig placed out in the summer season. So how do we use insects to estimate how long the minimum time a person has died? So if you get to the crime scene, let's assume what you're seeing there is a human body. Just assume that for a second. And then you see that there are several maggots already at the head area and in between the forelimbs. So what you do is you try to scan around the body and you try to identify the oldest. So what I mean by the oldest is the largest maggot that you can see. You try to look around. So mostly maggots are often in clusters. They call them maggot masses. So they feed in clusters because when they feed in clusters, they generate enough heat. So insects are very smart. They generate enough heat, and that heat can help them to last longer, to grow faster, and to feed faster and more. So you more or less look at the different clusters of maggots on the body, and then you collect more or less the largest size. And then once you collect the largest size, insects are generally poikilothermic in nature. What that means is that their growth, their development is dependent not only on the quality of the food that they are feeding on, but also on the ambient environment. So they draw energy from the ambient temperature. So in a situation whereby it is rainy, it is cold, their development will generally be retarded or slow down. But when the sun is up there and it's high, like in the dry season or in the summer season, they feed faster, they grow faster, and they are very aggressive. You understand that? So we need to factor in the ambient temperature, whether it is raining or not. We need to factor in the wind. We need to factor in the habitats, the environment, and all of that. So you collect the immature stages, be it the maggots, it could be the pupal casing, and then there are equations, there are steps you use to try to estimate how long the body has been there. So what happens here is that let us assume that it takes 24 hours for an insect to transition from the egg stage to the larval stage. Let's just assume that. It takes 24 hours for the insect to transition from the egg stage to the larval stage. And it takes 48 hours for the insect to transition from the egg stage to the pupal stage. So if you get to a crime scene and you see the larval stage of a particular insect, so there are different insects here in LASU, at least we've seen some of them, and there are different insects all over. So you cannot assume that one developmental process of a particular insect is the same as the other. So insects, they've got different behaviors, they've got different developmental time. So This hypothetical insect we're talking about. Let's say you found the insect X and it takes 24 hours to develop from the egg stage to the larval stage. So you come to the body and you see the larval stage. You see the oldest, largest larvae, for example. By collecting the oldest or larva stage, you identify the insect. So we've identified the insect to be X, insect X. And based on published literature, they've shown that it takes 24 hours for insect X to develop from the egg stage to the larval stage. And you find the larval stage of insect X on the body. So what that means is that you can then use the insect's developmental stage to say, because let's say, for example, Professor Adedeji published a paper and said insect X takes 24 hours to develop from the egg to the larval stage. So because Professor Adedeji in LASU or in Ojo area has done a study on insect X and it takes 24 hours, we are saying that, for example, we are saying that this body has been lying here for a minimum period of what? 24 hours. Do you understand that? So we are not saying that the death took place exactly 24 hours, but we are giving you a time frame based on the developmental stage of the insect. So from there, you can say, since we have got a time frame, then you can ask, who was the last person that visited the person? Who was the person last seen with? Who did the person call last? Then you can gather different people and different potential suspects, and then from interrogation, from CCTV cameras, from pictorial evidence, from testimonies and all of that, you can reduce the pool of suspects, and hopefully you can be able to identify perhaps who put the body there. At least the body did not just disappear there. Do you understand? So that is how we somewhat use the developmental stages of insects to try to establish the minimum time a person has died, or even an animal. So that's more or less the first step. So there are different insect groups. They've got different developmental stages, and there are insect groups that arrive at different times. So from my research, I have seen that some insects, when the body is fresh, they love it when the body is fresh. They will always be there. Within minutes, once you drop the pig carcass, you see them around like, oh, what's going on here? I love this. I'm interested to interact with the body. And then there are some insect groups. It is when the body is smelling, when it is stinking, when it is decomposing severely, that's when they want to come there. So that would lead us to the next slide. So that is called, so this is just, I did my study in Cape Town, and I used baby pigs to see how neonates, babies in quotes, would decompose over time. And then I compared the data with a full-grown 60-kilogram adult just to see if there will be differences. So this is a slide that shows the various decomposition stages of the 60-kilogram pig carcass that I showed you earlier. So we put out a pig carcass on the 15th of December 2022. That's in figure A there. You could see it's fresh, it's looking nice, it's looking it looks like something you can steal and take home and cook and eat for those that eat pork. And then within a day or two, about 3 days, that is what we get in figure B. And then after about 5 days, you get figure C. And on day 8 days after we've deposited the pig at this site, the pig has already transitioned into that stage. So figure A is the fresh stage, figure B is the early decomposition stage, the figure C is when it is in the advanced decomposition stage, and figure D is when it's more or less in the skeletonization stage. So aside insects, there are other procedures we use to estimate how long a person has died. So if you put a body under the tree, and if you put the body outside, or if you put the body in this room, and if you put the body outside, they might not decompose at the same rates. And the reason is that the temperature indoor might be cooler or hotter, depending on how hot it is, compared to the temperature outside. Also, insects or animals like rats, or animals like porcupines, animals like mongoose, or animals like dogs, if they have access to the body, they would speed up the process of decomposition and also compromise the integrity of the corpse. So in those cases, that could be misconstrued. There could be misconception by saying that, oh, it seems somebody chopped off the head of the deceased body, or it seems somebody created a wound. Actually, those things were caused by animals and not humans. So we need to be careful, and we need to conduct research in each characteristic environment. So let's assume in Lagos State, the medical school or the pathology office, they receive lots of bodies from Ojo area, for example. We need to do research in Ojo area to see how a body decomposes in Ojo area compared to Yaba, or compared to Epe, or compared to other areas, because different regions, they've got different sort of climatic variations that often impact on the insect assemblage that fits on the body, and also if the body is exposed to other vertebrates like rats, rabbits, and dogs and cats. So this is just a typical study here in South Africa in the summer. It took 8 days for a 60-kilogram pig this big to transition to what you see in the figure D. And here in Nigeria, I've heard that it took sometimes over 30 days for the pig to transition to this stage, based on some findings by Dr. Alafia and other colleagues in other parts. So it's key for us to know what's going on in a different region. So haven't explained to you all how we use the developmental stage. The second complementary approach we use is what we call the insect successional pattern. So remember I said that there are some insect that prefers when the body is fresh. There are some insect that prefers when the body is swollen. There are some insect that prefers when the chemicals, the fluids, the juices from the body are sort of seeping out. That is when they want to be there. So depending on the environment, there are different successional patterns. So they come in a predictable sequence. So once we know that insect A prefers to come when the body is fresh, insect B prefers to come when the body is in the early decomposition stage, and insect C prefers to come when the body is skeletonized, we've established a sequence. So that sequence of arrival, that sequence in which insects use to interact with the body, is what we call insect succession. Insect succession. So another simple example I can use is, when we know that, for example, I'm just giving a lame example here. We know that, for the first year we get a male, the second year we get a female, the third year we get a male, the fourth year we get and there's succession like that. There's a trend. So each environment, they've got their own specific trends. So if we can establish the trend here in LASU, and we see a human body, and we go there and we see insects A, B, C. From there, we can deduce that, oh, since we know that it takes about 5 days for the body to get to the skeletonization stage, and these insect groups prefer to come when the body is skeletonized, we could produce an estimate and say, based on this study by maybe Dr. Lafia and our colleagues and team, they've already established the sequence for this area. Then we can use that data. We are not telling the court of law or the prosecutors the exact time of death, but we are giving them a minimum estimate that could help them to what? To continue. There are some studies that have shown that insects sometimes do give very accurate information that is very close to the time the murder was committed. But we often say, for safety reasons, because if you go to the court of law and you are trying to show that this is when the death was committed, and a skilled lawyer who has got some forensic entomologists in the defense team would counter you and probe your question, and then your science becomes faulty. Is that okay? So this is more or less a successional chart. So just one information here. We could see that the first insect, Chrysomya chloropyga, you could see that it arrived on the pig carcass on day 3. On day 3. But then if you look at the last one there, you could see that the first appearance of that insect was on day 2. So different insect groups have gotten different arrival patterns, depending on the environment, depending on the weather patterns, depending on a whole lot of other factors. If a body is placed in a suitcase or in a Ghana must go back, of course, the body is prevented from the environment. So insect may not be able to access the body until maybe the Ghana must go or the suitcase is torn, or maybe there's a forced entry by someone, or maybe the wind, somehow damages some parts. So that's why we always say insects may not accurately give us the time of death, but they can give us an estimate. So maybe they only got access to the body after 6 days. What do you then say in that case? So if you just jump in and say that this is the minimum time of death without backing it up with scientific evidence or research, then it means that your presentation to the court of law is faulty. Someone who is very intelligent find gaps, and then of course take your file or your presentation and they throw it away. It's lacking scientific merits. So we need to firstly generate baseline data before we start thinking about using insect evidence in criminal investigation. So this is just some additional findings from our research. And so we found that ants, that we sometimes see, some species of ants, they are capable of creating injuries on a deceased body. So we need to be careful because sometimes someone would, in the process of trying to kill someone, they create injuries on the body, and that is inflicted by humans. But sometimes someone might kill a person and just take the body, and due to the fact that obviously killing a person is hard, carrying the body is also hard. You don't want people to see, you just dump the body somewhere. There might be ants around the environment, and these ants, they are generally omnivorous, feed on a wide range of things, microorganisms and all of that. So we found that some ant species can create injuries on the body. So if you look at some of the figures there, especially figure C, you will see some injuries, some black serpiginous sort of lesions on the body. So these are inflicted by ants. But as if somebody who is not knowledgeable, who is not aware, who does not know the literature, police officer or criminal investigator goes to the crime scene and sees a body with this sort of injuries, the first conclusion they might say is that this is caused by somebody. Let's look for somebody to arrest. But actually, this is caused by ants. So we need to first understand how does the environment, the insects, the animals around interact with a deceased body before we then decide to draw conclusions and say this is probably a homicidal case. Meanwhile, it is caused by ants, or it is caused by a dog, a stray dog around, or a cat, or a rat, or a rabbit. So we need to understand where. So we deployed the pig on day one, and I think within 3 or 4 days, we saw massive infestation of ants. Ants were on the body, and you could see in figure A and figure D, and with yellow arrows there. So those black things there, they are ants, and they were feeding voraciously on the ants, and this led to this sort of scalding or injuries or lesions that you see in figure B and figure C and figure E. And then sometimes too, we found that ants could bring about hemorrhage. So here we deployed the pig on day one, and after about 3 or 4 days, the ants were already feeding on the body, as you can see, at the region in between the forelimbs, the chest area. And then on the 4 or the 5, I think, we realized that that site where the ants fed on, blood was oozing out from that site. So people that are not experienced and do not know that ants are capable of doing this sort of impact, they could just jump to the conclusion and say, it seems this person was stabbed, or maybe there's some sort of stabbing, or there's some sort of penetrative trauma. But actually, this was caused by an insect. Do you understand? So that's why we need to understand what's going on in our environment. So when we used baby pigs, we decided to see what we saw on small pigs or small babies in quotes. Can we generate the same data? Are they similar or different? In the broad research story, we found some substantial variations between a small-sized pig and an adult-sized pig. And then we found that even on a very large 60-kilogram pig carcass, ants could also bring about the same scarring or the same injuries. And you could see it in a figure A there, where the ants were feeding on the external part of the right ear. And on day 2, you could see that whitish bite marks of the ants. And from day 3 to day 8, you could see a change in coloration of that same ant injury site. So if someone goes to the scene and sees that a part of the body of the corpse has got some of these sort of injuries or lesions, they could start saying, is it that there's a foul play here, or is it that the person that brought about this death sort of inflicted trauma on the person before death, which could further complicate the case and bring about more sentencing or more complicated sentencing. But actually, we need to know that ants and other animals can do this sort of injuries or lesions on deceased bodies. And almost coming to a close. This is the general workflow. So it is expected that when you go to the scene of crime where you see a deceased body, most times you will find insect there because insects are more or less like the most populous animals in the world. So it is likely that you would find insect. We generally say when you encounter that kind of situation, look for a forensic entomologist. They should be involved in investigative team because they know how best to collect the specimen. They know how best to preserve the specimen. They can, guide you on how to make sure that what the information you're collecting is valuable and is useful. In some instances, for example, in South Africa, we've got to train some forensic pathological officers. We've got to train some police officers, and at least from what I was told by my supervisor, and they've done some of those trainings. And you train police officers how to collect. You train police officers how to identify some of the frequently seen insect groups on deceased bodies around. So when they get to a scene of crime, because the forensic entomologist, maybe for example, Dr. Alafia, she's in the class, she's teaching, or she's in the laboratory doing research, and you find a body somewhere in Ekpe or somewhere like Yaba that is very far, and obviously she can't be everywhere. So the police officers there can say, "Oh, doctor, we found this thing." By virtue of the fact that maybe she may have trained them on how to collect, how to store, and how to transport the specimens, they can proceed with the work, and then she can then do the administrative and scientific aspect, depending on her schedule. Or if there is a trained forensic entomologist in the police, then that could make life less complicated because, of course, the police will just go with the entomologist who is also a police officer, and then they can analyze and interpret the insect evidence. So this is just like a workflow. You collect the insect from the deceased body. You collect two groups. Some you kill them almost immediately, and for some you have to breed them. You give them food, they eat like a normal human being would eat, and then they grow. So when they grow, it becomes less complicated to identify them. So we often call that companion sampling. You take some for preservation and killing and PMI, minimum estimation, and you take some to be sure that's what you are trying to identify. Because if you collect the egg stage, it's hard. It's almost impossible for you to know what species you are dealing with when you collect the eggs. So sometimes you take them to the lab, you give them food like a rotten liver or meat or fish intestine or whatever. So they feed on that, they develop to the maggots, they develop to the pupa stage and to an adult. So when they grow to an adult stage, you identify them using published morphological descriptions of these. And once you do that, you often do that under a microscope, and then you compare. Like I said earlier, when you know that it takes 24 hours for X species to develop to the larval maggot stage based on published studies, you then do the comparison and say, oh, I collected S species from the body, and someone has shown in Nigeria, in Ojo area, that X species, this is the amount of time, depending on the temperature, for it to develop to this stage that you collected from the body. Then you can say that the minimum time the body has been lying there, the minimum time the person may have been dead is this amount of time. And then you look at other factors like weather, you look at clothing, you look at wind, and you look at season. So when it is very cold, most insects are docile. They are often not active. But if it is hot, like in Nigeria, you will always find insects almost everywhere, even in your house. Insects are trying to obviously drag the house with you, and depending on those variables, there are other variables that could confound your sort of accreditation. But we need to be aware, depending on the variable. In South Africa, we have realized that the presence of clothing on a deceased body would influence the decomposition rates and pattern compared to someone who is nude. We have also realized that if a body is inside compared to a body that's outside, obviously the one outside will decompose faster because that could be exposed to wild cats, obviously exposed to the sun. There are some other variables that could feed on the body and also speed up the process of decomposition compared to when the body is inside. But if, for example, the body is inside, the door is open, there are animals or dogs or whatever around, we do not know what can happen. So there are various factors. There are trauma. Trauma could also influence stab wounds, gunshots could influence the process of decomposition because they create opening in the body. Insects would capitalize on the openings. They will lay their eggs and maggots there. Then the maggots would have access to the body. They start feeding on the body immediately. and that speeds up the decomposition. Some other studies have shown that those openings actually do not have an impact, or it in fact reduces the rate of decomposition. So we cannot just wake up one day and decide to say we want to use the information or data from Kaduna or from Kano or from Enugu to try to disentangle cases in Lagos. We need to generate specific, location-specific and region-specific data to use in criminal investigation. So that's what we always advocate for. And we can't just go out there and say we want to do research. If you're doing forensically relevant research, we need to look at the trends of death. So if we see that in Lagos or in this area, most people die by gunshot wounds or stab wounds or machete or intoxication, then we can do research in that regards to say, let us now simulate a crime scene where the animals are terminated or euthanized ethically, and then we look at intoxication, we look at gunshot effect, stab wounds, we look at clothing. So there are a wide range of variables that could interfere with the decomposition rate. So in summary, I hope that you guys may have gotten a bit of appreciation of what forensic entomology is and the various concepts with respect to time since death estimation, and also the different ecological categories that of insect that interact with the deceased body. And I think that will be the end of our sort of presentation. And I'm happy to entertain questions and comments.
Good afternoon, sir.
Absolutely.
Okay. Based on the lecture you just received, sir, can it be said that the knowledge of this etymology can only be used to ascertain or to determine when death occur, or can it be used to determine the cause of death? If so, how?
So thank you, sir. Thank you so much, Dr. Aditme, for that very insightful lecture. So just like he said along the line when he was presenting, he actually said that you could use it for various means, and I highlighted some of them. For example, you could tell the place of death. Aside from telling us the time, you could tell the cause of death, and sometimes also tell some other situations surrounding the death. For example, if you are talking about cause of death, cause, we could use, there's an aspect of forensic entomology which is called entomophorensic forensic entomology, whereby if somebody dies as a result of intoxication or due to some drug-related cases, collecting the insects that you find around the body and rearing them, looking at the rate at which they grow, and even identifying their tissues, some things in their tissues, some of those toxins in their tissues, we can actually tell if that was the major cause of death, or if by chance there were some other things that had led to the person's death. Let me just explain further. Like if you find the toxins inside the tissue of the insect, and then maybe you just found the body, you don't even know if he has taken some toxic substances, but the person is dead, and you find these toxins inside the body, the tissues of the insect, it could lead us to say, especially in the what quantity of the toxins is inside the body, to tell you some things about the cause of death. And he did mention some insects that you find in specific areas as well. Now, while he was showing me some of his slides, we have a certain species of blowfly where you find some of them in the rural areas, you find some of them in urban areas. So if by chance the body had been moved, if the person died somewhere and somebody's trying to cover up, took the body from somewhere around Ikorodu or Epe, brings it to Ikeja, for instance, And you're seeing insects that are supposed to be found in rural areas, on the crops that you find on the urban. That would give you some inkling into maybe where the body had been placed, if you suspect that the body had been placed. Let me also give you a very good example of how you could use insects to determine maybe cause or time of death. Now, there's this case of a woman whose body was found at the beach in Sicily, Italy. Now, the woman's body was found on by the beach side, but then they actually been looking for the woman before they found her body. Now, questions arise. Who was the last person that was seen with the woman? When was the last time they had contact with the woman? And then they found out that the last person that was seen with the woman was a certain man, an influential man in Italy. So on going to the man's house to make inquiry, they said they found they couldn't find anything. But the only thing that now led them to know that it was the man was that because the man denied, he said he didn't see the woman, he has never been close to her and all that. But on the wall inside his house, there's a mosquito. Now that mosquito, we also, like he talked about the ecology of the insect, is known that the distance in which mosquito could fly. It couldn't fly so far away from the man's house to have gotten to the beach. And on the wall, the mosquito had been killed. So the entomologist picked up the mosquito, looked at the tissue, found that the woman's blood, the DNA of the woman was on that blood. Of course, then after further interrogation and all that, the man confessed that he was actually the one that killed the woman. The woman was inside his house and he went to dump the body somewhere. So that again is just a good example of how you could use the insect to tell. I hope I have that clearer.
Thank you very much. Please, I may want to know or ask, such insects, can they be classified during this experiment? Like the colored, the striped, um, um, the blue fly and the other fly and the class of mosquito. So when you like this buzzing fly that will just come and if we can, if we are able to identify such flies, we can now know that yes, something is wrong somewhere. This news is coming to let people know that something was really wrong. Could they be all classified according to their species? Thank you.
Yes, definitely. So one of the key um, processes, like if you look at this slide now, I think, thanks. This is Califora vicina. So this particular fly is cold adapted. What that means that it's always abundant when it is cold. When it is very cold in the winter season, that is when you see this fly. So before you even think about using insects to try to determine the cause of death or the minimum time a person may have died, we need to first accurately identify the species, and
And deposits its egg and the maggots on the body and all of that. And let's say along the line, the body was conceived or was kept in a place where there's no access. And maybe after a while, a different insect group arrives on the body and lays its egg. We would see that some are older than the other ones. So that's why I said when you go to the scene, often try to look for the maggot or the larvae, those regulators that is the oldest, the largest, or the longest. In some cases, you will see maggot and you will see pupa, those brown boxes. So intuitively, you should know that the pupa cases or the pupae of the flies, that's the oldest stage and not the maggot, because some of them have transitioned into the pupa stage. So we collect the pupa stage and we also collect others. So when we use that, we can then create different timelines to say, based on insect X, this is the timeline we are giving, maybe 2 days. Based on insect Z, this is the timeline, maybe 5 days. My report will then say, generally, the time the body may have been deposited in that environment is between 2 to 5 days, based on these two insect groups. So you can decide as the investigator to start your investigation from day 2, before the body was discovered, or from day 5. But now you've got a range or a clue as to the possible interval. Of course, there's a joke in our lab. We say that you cannot ask the insects when it arrived on the body. Obviously, they will not respond to you because they can't talk. But by virtue of their size, their developmental stage, you can create a timeline that can serve as investigative leads for other investigation. So in fact, some cases you get multiple species, 3, 4, 5. So that's what you do.
If I might add, of course, when you get to the body, you are looking out for larvae. You cannot decipher which larvae is. They all look alike, especially if it's fly. If the diphtheria larvae, they all look quite similar. So you can't really tell by. That's why you need a forensic entomologist that will collect the larvae, rear it to get to the adult, and then we can get some information. Now, talking about determining the exact time, like you said when he was presenting, we normally do what we call minimum post-mortem interval. We can't tell the actual time, but we can give you all the things that will lead for you to unravel the case. So, of course, the forensic entomologist uh, most times would give you a fine estimate of time range and not be too specific as to say. Of course, you can be as specific as saying this body, from what I'm saying, is 3 days. I can see that the insects have transitioned into the pupa stage. And of course, when they even get to the pupa stage, you'll find out that the insects are living. They are leaving the body because they do not want it to be moist, as it were. So they tend to move away from the body, and with that, they also find them in some other places close by the pupa of the insect. And sometimes, if you find some other species of insects entirely, like coleopterans. Now, we talked more on those common ones that come very early. There are some that come also much later that we could also use to determine. And with all the information, we have to put all this information together before we give you a specific time. Yes, there's somebody else here that wants to.
Good afternoon. Our forefathers, because there are some times when the body, someone will be killed and buried, and you continue searching for such a person. Our fathers, there are insects. When maybe they are hunting for animals in the bush. When they see such an insect in an area, you will actually know that there is a dead body around. that can at least lead someone, a layman, to such a thing. Sometimes policemen will be looking for where someone until you see a suspect taking you to that place. Can you shed more light on this?
Okay, I think that's a very fantastic question. I think so. We can approach this in two ways, and I'll throw a bit of information on decomposition, depending on the ambient temperature and the weather generally. The body generally starts to give off what they call volatile chemicals. Generally, they've got a very sensitive smell to these volatile organic chemicals. So they are able to detect these volatile organic chemicals. That's why you wonder, for example, if you've got something rotten in your dustbin, like you just realize that insects are already there. How? So they are sensitive to these chemicals. They are called VOCs, organic chemicals, or epineumones. So once they see that, the insects would arrive on the body. So let's assume insect A arrives on the body first. Insect A will start feeding on the body, and it will deposit its eggs, and the maggots will start feeding. The maggots, together with the odor coming out from the body, they secrete a unique kind of cocktail of odor that goes off also. So that signals to other insects that actually there's food here. Guys, come. There's food here. So from there, you see a fly. From one fly, by the time you go there in the next 5 hours or 10 hours, you see hundreds, you see thousands, you see maggots all over the place. So the body has got endogenous bacteria and enzymes that start decomposing immediately death has occurred. And along the line, when the body is swollen, the bloated stage, the body will obviously now burst open and start giving off more of this odor. And then obviously as humans, we say, how do they smell like this? So before even humans detect that smell, insects are far more sensitive to that. And by virtue of the fact that obviously there are insects buzzing around in a given area, somebody who is intuitive enough can say, there's something going on here. Why am I seeing flies? Why are flies so abundant here? Then from there, you can start asking questions and start looking around. So that's one way insects can be used. In some countries, in some advanced countries, there's got what I think Nigeria has got that canines, where they use dogs trained to detect the volatile organic chemicals, the decomposing smell from human cadavers. So they train these dogs over time, and these dogs are extremely sensitive. So if you go to airports, for example, you see some dogs that are trained to smell cocaine and those illicit drugs. So the same way they train dogs to be able to identify these smells. So instead of you focusing on insects, there's what they call human cadaver detection dogs, HCD. So they are able to detect the smell. So the insects, the dogs will train you or help you to identify the source, the site. So if you see that the dog is somewhat digging into the ground, then the dog is obviously telling you that there's something here. You understand? So the same with insects, when you see that there are lots of insects buzzing around in this area, which is unusual, especially if it's an environment where people often pass through, then that should tell you that, yeah, what's going on here? Why are insects here? So if I come into this space and I see lots of blue flies and flesh flies, I'll be worried. So is either there's a dead rat somewhere, or someone has kept food for a long period of time that is now decaying and smelling. So then I'll start asking questions. What's smelling? Or why are these flies here? Is there an entry point through which they are coming into this space? Or is there something that they are sensitive and attracted to? So these are how we can then take leads to be able to identify the corpse or the buried corpse or the body parts, maybe the head or the limbs or whatever. And then you can take it from there with the police and all of that. I hope that provides clarity.
Just to make it clear, there are some insects that you would see that you actually tell if there's, and that it also depends on the size of the decomposing animal. So if it's very small, the stench might not be strong enough to attract them. Or if it's as big as a human, you would find a certain group of insects. We call them, the first group that usually comes are the necrophagous insects. So the Caliporidae and Sarcophagidae, these it's just some names of some of the insects. If you see them, you can actually begin to be like curious, like what is happening here? these insects are not supposed to come ordinarily.
So that's why if you open, let's say you are cutting your fish at home and you start taking out the guts, the intestines, of course, along the line you see some flies, and then as you're cutting the fish, obviously you start swatting because the flies are now sensitive to the volatile organic chemicals coming up from the fish that you are trying to cut, wash, and of course cook, and all of that. Same concept applies.
So do we have some more questions? Okay, I think there's one last one.
The last one. I'll go to Fancy now, right? Fancy.
Please, I just want to clarify something. You know, when you were talking about one experience about somebody that was dumped around the beach, and then when investigation was being carried out, that a mosquito was found in the room, and when the mosquito was killed, the blood was taken maybe to the laboratory, and then we found that that was the blood of that very woman. But let me just say this, because I know sometimes our human beings, sometimes we can make mistakes. All these, when we say forensics entomology, all these findings at the end of the day, the final result, could it be something like, is there any practical means of getting the best result, or is it based on speculative knowledge that maybe sometimes, just as you said when you get it, you said you cannot ask a fly how long you have been on the body, but when you see the developmental stage of it, you cannot tell even the fly how to develop. The fly can decide to develop based on how it feels. Just like human being. The other day I went to the laboratory I decided to go for all-round check on my body. So when after taking my body, my blood, and then the urine they asked me to bring. So when I was taking it into the laboratory, there were many. They gave me a particular plate. Inside there were many urines and many blood. And then asked me to put it there. I said, how will I put it here? How will you know my own? At the end of the day, you will not give me a result that is someone else. So I need a plain plate that will contain my own. So sometimes that very blood in that very mosquito, after 2 days, scientifically, will that blood still give you the best result you need? Or because I know after 2 days or some days, that blood will change. So if you take it to laboratory, will it still give you the perfect result you need? And then at the end of the day, when you are checking, or you want to, and maybe as a forensic entomologist, you are now deciding when the body or the person was killed based on the developmental stage of the maggot or the insect. Now, can you get the perfect result there, or are you speculating based on your own knowledge?
Quite an interesting one. Now, to start with, I'll go back to what we said earlier. What we are determining is the minimum post-mortem interval, which means we are trying to tell you if it's talking about the cause of time of death, the minimum time, and sometimes you could call it the period of insect activity on the body, how long insects have been able to feast or to lay their eggs on the body. Now, broadly speaking, now there are some insects that would lay larvae, just like you were saying. There are some insects that will come to the body, and what they are laying is not eggs, but larvae. Now, so it's only the forensic entomologist that can tell you if this larvae is that of a Sarcophagidae, which has been known to lay larvae and not eggs. And of course, that has speed up the time. Or if it's one that is going to be laying its egg, and the egg has to transition into the next stage. Now, talking about whether we are making speculations, it's not just speculation. It is based on simple normal logic, based on studies over time, based on a lot of not just what we say hearsay, and then most of the time the forensic entomologist is just leading you to something. That is why I gave the case of the woman. It is the woman's DNA that was found inside, not any other thing. Now, is it possible for the mosquito to carry somebody else's DNA and for it to be what's it called, assumed to be somebody else's? So that's not possible in that case of the woman. it was our DNA that was on the blood. And so, of course, you know that for our DNA to be in the mosquito, it means the mosquito must have fed on her. And the mosquito has been known, like I said, not to fly such distance from the site where her body was done into that house. It means that she was within that vicinity. And we don't use all, now we're talking about admissibility in court. You cannot use the old evidence. It's just to lead you to do further investigation. You are not using the forensic entomologist cue totally. Now you need, that will raise some questions, would lead you some other things that will now make you to widen your, maybe your scope and your area of research, or for you to be able to tell that, oh, this and this, it's probably what is happening. It's not that we are making speculations. I would like allow Dr. Adetime to also add.
Yes, I think Dr. Lapia gave a very fantastic one. And just to add to that,
so before you add, I can see that the entomology is just a lead. In criminal investigation, if a suspect says, yes, I forged the document, you will not just stay there and believe that he has forged the document. Because by the time he gets to court, he can put up forward that, no, it was based on duress. For you to be able to prove that, yes, he forged the document, you still need to send those documents to forensic analysis for them to prove or confirm that, yes, the documents were actually forged. So in forensic entomology, by this, it gives you appropriate time when you get to court. It means you still need to carry further investigation to confirm that what you got through the analysis of entomology. So just stay there, yes, if they get to court within 5 days and you accepted that within 5 days, further investigation still needed to be carried out to corroborate what the results you got from the entomologist. Thank you, sir.
Yes, I think that's fantastic. Like I said, it's we these days we advocate for multidisciplinary research. So if you're investigating a case, because you found an insect there, doesn't exclude the forensic pathologist, the forensic toxicologist, the forensic odontologist, the forensic toxicologist, anthropologist to come and look at the body. Because it's now all of us who sit together and then we write a report. So in South Africa, if I'm involved in a case with my supervisor, we put up a case which is called a forensic entomology report. Then the leading pathologist, based on the South African law, the leading pathologist will now ask a forensic toxicologist if there's suspected toxicant or poisoning. A forensic anthropologist, they will write their report, an odontologist will write his report. At the end, we put all the reports together, we sit down together and make meaning or reconstruct the situations or circumstances that led to the death of that person. And also the health status of the person. So if you're looking at a case of intoxication and we're looking at cocaine, it could be that that person may have died of cocaine use, but it could be that that person was a cocaine addict. And sometimes some of these drugs or these medications, they stimulate the development of insects. So if an insect feeds on a cocaine-infested tissue, there's a probability that they will go differently compared to when they feed on a tissue that's got no cocaine. So these are variables that only the forensic entomology cannot sort of disentangle. So that's why we say we are collaborating. And we don't just come up with speculations, we come up with results based on scientific evidence. So we don't just wake up and say because of this and that. We use scientific procedures and steps to try to establish what we are trying to establish. So I think with that, we've come to the end of this sort of phase and we transition into the next. So I'll hand over to Prof.
Thank you very much for an insightful and detailed presentation. Honestly, you did more than I expected. I mean, the two of you did more than I expected. And I believe that with this first experience, we're going to need more, because it's not just for us to have one-time experience and feel that we are equipped enough. We need more. We will do more. The next phase is for us to go to the crime scene. Now, there are two ways. You can choose to be here because of the number. If we go there as a crowd, it will not, there'll be noises, there'll be doors, it will affect a lot of things. But if you stay here, we are going to be showing you what we are doing over there on the screen, and it's going to be audible. So I want to recommend that we stay here, and then the multimedia, myself, okay, multimedia stays. You will stay too. Eh? So you stay. So I'll use my. You want to be there with your camera? Okay. If you go there with your camera, it means I'll go there with my laptop. Okay. I'll go with him. We will be showing you what we are doing here on the screen here, and it's going to be clear. So we will leave with Mr. Paoli, you are here now. So we leave with your speaker will give us feedback. Let your speaker give them feedback. Yes. Your speaker will give them feedback, and then we operate from there. Are we fine? So that's what we're going to do. So you card then coach.
Now let us make use of this one in case I don't know. We don't ask questions from here. If they ask questions from here, then we'll be able to respond from there. I'm just asking.
But you don't ask questions. You're just going to listen. Check out your phone. Check out your phone.
So I go to connect now. Just have to
So you will hear from the speaker. So two people with 2 cameras to follow us.
some of us can stay there.
No. I only need 2 cars. I don't want you to leave here. That's why I'm not calling.
Madam, I work now. Are you enjoying this vicinity? Yeah, the environment. Yeah. Oh, compared to that Falun. I said this area compared to that Falun. That's what I'm asking. Yeah, very cool. Like, yeah, it's polluted. Noisy, really. No, you are used to so many people. Yeah. You are used to so many people in Falun. But the expenses is low compared to yeah.
Are you using this?
Yeah, you said that you
This is mine.
okay, zero.
So I'll come back.
Yeah, yeah, you're right. But they are more cheaper here compared to if I'm here in a week, I don't spend that much compared to when do you not shop? Truly, they are very used to that market. For me, it's only chewing gum created out of 36 star. If I was expecting pound. Hope you're not bored here. Hope you're not bored here.
I am here.
Yeah,
Now
accepted one now.
you now you make me spotlight
Okay.
when I'm there. What to do? Coast. Where's Coast? Is it Coast?
Is it? Make us.
So when I'm there,
All right, sir.
Baba, you are not doing Baba.
No, na rubber. Na rubber.
What? Thank God. I don't want to go swim today.
What is not? Now that's something to say in the challenge.
To be clean life. Sorry, Mama, it's only here.
You are not gonna.
Yeah, don't know that. That boy will not come. That will do it. Is that it did not come today?
No. But you just follow them out.
Only pull them out. When you have to sit down, I will see.
We buy dog chain.
Dog chain, thank you. But I'm looking for 1 month since money I have not seen. You see, otherwise you see your government exams. And the serious students, the exam day. See what? On the exam day. Guys. Olórin. Now what you just like you it's another you.
Abolo. Abolo.
Those mask, wọ́n rà kan.
Ilé Yorùbá ti rí ètò ọmọ yìí lọ́dọ̀ fún ẹ̀rọ títí lè lọ sí fún ẹ̀sìn. Why is this? That's all we want to do. See, God is very good at the family. He has
Bros, are you still online?
Yeah. Yes. Òun kọ́ lẹ lù lẹ́ẹ̀kan.
Mo ti parí HDMI mẹ́rin nísìnyí. But tá a bá connect ẹ̀ sí kìíní yẹn ó máa rí. Nǹkan tó ń fẹ́ display látọ̀hún yẹn kan. Ẹ bá mi fi sí ìdí ibẹ̀ yẹn ó máa rí. Sorry, let me remove.
Okay. Yes, sir. Yes, you know that. You cut down wrestling talk. Say that so we work in so I can start with that. Start taking one second of the like that. So no like that. Then you always express in your so that's why you laugh. No, the world can hear you. Can you? Well, my life can
The practical is about to get started. They are trying to figure it out. No, just about to. Well, you can still go online. You can still go online. Go on YouTube. What's that? I didn't know it was a DK one. You are using for the expect. Mm-hmm. That's why they killed it a few days.
They said you mute
Okay.
Before last one, all those videos don't like to mute.
Yeah, it's done.
Is that the last one?
All right. Good afternoon. I hope you all can hear me. If you can hear me, maybe just put a thumbs up in the chat or just react to this so that we can know, or if I need to speak louder. So we are at the simulated scene, the simulated crime scene. And what we are trying to portray here is how we collect entomological specimen. Of course, if you look, this is Dr. Alafia, the forensic entomologist at the Lagos State University, and I'm Dr. Adeyemi Adetime, also a forensic entomologist in our lab. So she's fully dressed in our appropriate PPEs, which is the personal protective equipment, and she would sort of be the one collecting. So behind us there, not sure you can see it clearly, but behind us, we've got the carcasses that we'll be using to somewhat simulate the whole procedure. So here you've got your sweep net, which is what we'll use to collect the adult flies that are flying and hovering around the body and the scene of death. And then we've got an insect cage here. So what we hope to do today is to collect the adult flies and try to transfer them into the insect net, and then we take the insect net to the lab where we euthanize them, or we literally kill them by freezing. And that's the method we would hopefully use. And then we've got our vials and our collection tools here that we'll use to collect the insects. So we will be heading over to the deposition sites of the carcasses, and then we'll show you how to look at the body and regions where you need to collect the different sort of insects around. So I think firstly, we'll be starting with the adult flies, because once you start approaching them, what you do is you take photographs of the site even before you approach them. So there are standard procedures. So here we are not, of course, going to cover the whole scope of the whole standard procedures, the labeling of the scene and all of that. And we'll be majorly focusing on the collection of the entomological specimen. And during the course of the lecture, you will see that forensic science requires a multidisciplinary approach. You require everybody on all hands on deck to collect the specimen, and that's why you need different experts. But for today's session, we'll be focusing on the entomological specimen, and we'll try as much as possible to give you guys clear views of the carcass and the insect that are feeding or on or around the carcass. So yeah, thanks. And in the next couple of seconds, we'll approach the sites where the carcass is being deployed. And Dr. Alafia will be showing us the ways that the insect are being collected. Thank you. Okay. You want to follow? Hmm? Adult fly. So what we do is we take photograph, and then before, of course, he interacts with the body. And then in this case now, we've already enclosed the pig carcass because of the animals that are around. So Dr. Alafia is going to expose the body now. So if we look at the pig carcass, can you move closer, sir? That's okay. So we'll see that the body is somewhat swollen. So the body is currently bloated, and it is in the early decomposition stage with lots of adult flies, and some maggots are already around. So what we will do firstly is to collect some of the adult flies to give us a sense of the insect that are around. So Dr. Alafia is doing that now. What about the yes.
Okay.
So these are some of the adult flies that have been trapped in the sweep nets. And what she's doing now is that she's trying to transfer them into an insect cage where they will be kept alive. And then when we get to the lab, we'll kill them by freezing them or euthanize them by freezing them. So what she's currently doing now is she's transferring some of the adult flies that are hovering around the carcass, and she's transferring them into an insect net to keep them safe as representative individuals. You can give me this. You can bring the insect net closer. So, not sure if you all can see it, but you'd see that there are some insects that have been collected and trapped in the net. So this will keep them here, and then we'll have some representative um, insects for us to help us guide and direct us on the insect species that actually deposited its eggs or larvae on the carcass. So this is just an example of how we collect the adult fly. So what we'll do is we'll move closer to the carcass, and we'll show you the areas of the body you need to check to collect the eggs and the maggots and other crawling insects around the carcass.
Look, here, if you look closely here, you find this whitish marking. These are eggs of the of insects. Now you don't know which fly it is, so the best practice is to collect them and then transfer them onto substrates where they will be reared to get to adulthood. So you just collect some of them. That should do for now. Please, Dr. Laka, talk on the wound. Yes, so if you also look at now, this particular animal was shot. So what you have here is the bullet wound that was created. But then you can also see that there are some other ones on the body. We did say that some of them, of course, you can see these ants now. These are adult ants, probably Criminology, and they do make all these kind of lesions on the animal's body. So this should not be taken as some other kind of injury on the body. It's possible it has been made by some of these ants. And so then the ones are those maggots. Yes, so what we have there are maggots. They are maggots of basically diphtheria. We can't tell which one it is. So I'll just collectively say they are diphtheria. So we, like we said earlier, we have to look for the larvae that are the biggest kind of. So you can see they are of different sizes. The best practice really is to collect from different orifices into separate vials. So I'm collecting somewhere close to the ear, and then I will label appropriately that this was collected from the ear, this was collected from. So what we have here, these are some of the adult, the giant larvae, we have now you also are expected to measure the temperature of the maggot mass. Okay. And then you could see there are some smaller sized larvae here compared to the ones I picked. Very small. So they could be of some different type of insects, what we have there. And then if you go towards the oral cavity, you would also see openings that there are some other kind of now, because there are different species that like to inhabit different places, it will be best to collect from there to labels with special labels of the insect that the larvae from where it was collected. So with that, you could also do scoring of the body. Aside from using the insects, there are some things you will look out for that could score the body as to is it still bloated, has it gone deflated, and then some other. If the body is still as it is now, it has gone through. It's getting to the advanced stage of decomposition. So these are other things that you could look out for at the scene of the crime to understand what is happening there and what kind of insect is interacting with the body as it were. When we get to the lab, some of these insects you have collected, usually we have a certain number of sweep. We could have 5 sweep. What I did was just once. You sweep, and then so you can get a good number of different species of insects that are present at the scene. And then by wearing the larvae you have collected, will also give you a closer view as to the insects that you can find around here. So of course, now there are also crawling insects which are not like these ants. They are opportunity species. So what if you stay here for long, you see some of them picking up larvae and taking them away. So they could also affect the postmortem interval. If there are more ants that have taken away larvae, it means the mass will be as hot as the temperature around the egg mass, and that will reduce the rate of development. That's the insect. So there are lots of factors that we could also want to consider by looking at the body of the decomposing carcass. But I think we are good for now. So I'm going to cover it back. Now, the major reason why we are covering is for scavenger, because we have some animals to come out.
Okay.
Okay.
So, what we just looked at now is the pea carcass that was shot, and you could see already that there are hundreds, if not thousands, of maggots that are already feeding on the carcass in the oral cavity and in the region around the shoulder of the pig. And you could also see ants. You could also see several adult flies that are moving around. In fact, we just saw a beetle now to show you that at different stages of decomposition, there are different insect groups that, of course, will be attracted to the corpse. And actually, where she showed you that part of the carcass where you've got that thousands of maggots feeding there, that is the site of entry or exit of the bullet wound. So that could also tell us if there is penetrative trauma, because insects would want to lay their eggs in an exposed area because they believe that the eggs will hatch and the maggot will immediately start feeding on the soft tissues of the body. So this pig was killed by being shot, and the other pig will be going to now, still replicating the same procedure. We'll collect the adult flies using the sweep nets, and then we'll collect the eggs if they are present, the maggots, another crawling insect that are visibly present on the pig carcass. So we're moving on to the next pig. That pig was killed by exsanguination, and we'll be doing the same, replicating the same procedure in the collection. And then when we get to the lab, that is when we do standard procedures and all other activities to preserve the specimen and to process the specimen.
Okay. So this is what we have tagged as E2. Okay. And like you have rightly said, it was still there is evaluation. So we just want to look at what stage of the composition it is. And of course, taking in consideration environmental temperature and all the like. We can. Okay. So just like the other one too, you can see the orifice, all the opening from the neck region, from the mouth region, as well as on them. Yeah, unless you look closely, you can't find much of the eggs deposited on the body. But then most of the time, usually at the back of the ear anyway. So you can look for eggs, you collect the eggs, and then also look for the big maggots, like we said. So there are very tiny ones, of course, there are also some much bigger ones which will be collected into variable bottles. And then looking at what we have here, we have a large diversity of insects here already, diphtheria, sarcophagidae, and some other ones. So we would collect and also keep in the cage for further analysis. Yeah. Already from
All right. So yeah, so I believe that I understand this for now might be a lot of information to process, but this is just to show you, what happens in a typical situation where you find a body that is lying or being deposited by somebody in an environment, within your environment or elsewhere. And then obviously as a forensic entomologist, you come to the scene, you take photograph, obviously with your full PPE like Dr. Alafia, your face masks, your gloves, your lab coats, and just protect yourself as much as possible so you don't also contaminate the environment. And then you collect your adult flies using your sweep nets, and then once you're done collecting your adult flies, you move closer to the corpse or the carcass in this case. You collect the eggs as she has previously shown us on the first pig, and also you collect those crawling whitish things, which are mostly the maggots. So generally you look for the largest one, but in some instances, they are of most times or sometimes of the same size. So you collect a representative sample, you put them in your storage container, label them properly, and make sure that every single thing is well labeled. The time of collection is also well documented, and the same demographics is also documented. And then from here, normally you would move to the lab where you then start processing your specimens. So things like we preserve the adults, we kill them and we preserve them using standard procedures. The maggots you preserve, you kill and preserve some, and then you also feed some, you give some food sources like beef or liver or something like that so that they can grow. And when they get to the adult stage where they fly around or they move around, depending if they are flies or beetles or ants or whatever, you can then successfully identify the species. So that can now help you to determine the species that colonized and deposited their eggs or larvae on the pig. And at the same time, you can then start drawing your reports. So this is just an overview of what forensic entomology entails. And we've just simulated one that was killed by gunshot, the first saw, and the second one that was killed by ensanguination. And looking at that, we could already see that there are thousands of maggots that are already feeding on the body. And if we come back here, maybe in the next 5 days or so, we may potentially find just the remnants of the carcass here. So this is more or less like an overview. And of course, as you progress in your studies, if you are interested in forensic entomology, you want to become a forensic expert, entomology expert, then you may need to do your MSc, you do your research, and you do your PhD, and of course, you continue from there. So this is the end of the field simulation, and I hope you enjoyed the session. Bye for now.